Sounds good. And Robert's here. Hey, Robert, how are you?
Good. How's it going?
Yeah,
Hi,
great, great.
hi, Robert.
Nice to meet you, Marco.
Nice to meet you. Likewise.
Okay, so we'll just let people start coming in, and then we'll start probably right on the dot at 8 o'clock. Um, yeah, I guess the format basically is, yeah, 30 minutes of like questions that I have, just like stuff going over Rubedo and maybe some of the early days of the
Mhm.
company. And then afterwards, we'll open up halfway through at 30 minutes. We'll let people ask questions and then we'll just wrap it up. Should be just like an hour. So, yeah, so thank you for agreeing to come on. I know you're probably busy, but this is really great to have you.
No, of course, I'm excited and happy to be here. Thank you for the invite. Actually, I really like the initiative, so happy to give my contribution as much as possible.
Yeah, it's our pleasure to have you. Okay, so it is 8 o'clock. Robert, are you okay with the recording?
Yeah, I think we're good to go.
Okay, so I guess we'll begin. So, hello everyone. We're here with the Longevity Biotech Show. Today we have the great pleasure and honor of having Marco Quarta from Rubedo Life Sciences here with us. Rubedo Life Sciences is a senolytics company that targets senescent cells. And yeah, I guess just a couple of housekeeping things. This show is being recorded. So later on in the show, when we invite people up to ask questions, if you come up, that means you're consenting to us recording, using your audio in the recording and also your profile photo in the video that we upload to YouTube. Okay, so without further ado, maybe we'll just start with some basic introductions. I'll go first, then Marco, and then Robert. So myself, I'm Nathan Chang. I'm the founder of Longevity Market Cap and the editor of the Associated Newsletter. Basically, it's a once a week roundup of all the different developments in the longevity biotech industry. I'm also the founder of a Longevity List, which is a website where people can find jobs, companies, and investors in the longevity biotech industry. And then, yes, I guess I'll hand it over to Marco.
Thank you, Nathan, and thank you, Robert, for the very kind invitation to talk today. It's exciting to contribute to this, great show. I think it's really good for the field and to promote all initiatives around longevity therapeutics. Um, so about me, Marco Quarta, as you mentioned, I'm the CEO and co-founder of Rubedo Life Sciences, and I've been let's say in the longevity regenerative medicine space for a very long time. Prior to that, I was at Stanford directing my research team there, focused on aging regenerative medicine. Prior to that, I did my postdoc at Stanford with Tom Rando, one of the leaders in the field. So it's the Tom's lab is where the idea of the parabiosis was launched, right? And if you're not familiar with that, is this experiment, famous experiment that showed that you can put together, like combine surgically, an old mouse and a young mouse, so they can share the blood. And what they found is that the old mice were able to become younger from a biological standpoint, rejuvenate and regenerate better and recover functions, suggesting that there are some rejuvenative factors in the young blood, right? That then further studies show that this is true, and there are even companies who build upon this idea in the longevity therapeutic space. But I mean, so anyway, that's where I did my postdoc, working with Tom for years, and we still collaborate. And as uh, my former mentor in the space. Uh, in general, I have a PhD in aging in neuroscience, degrees in biotechnology, bioengineering, working always in this around this idea. Um, and as a scientist, then as an entrepreneur, Rubedo Life Sciences is not my my first experience. I started other companies before back in Europe and here in the Silicon Valley. So more recently, I also co-founded another longevity therapeutic companies here in the Bay. It's a spin-off of Stanford called the Turn Biotechnologies with my friend and colleague, also faculty at Stanford, Professor Vittorio Sebastiano. And that was a different story. We showed in that case how in a transient epigenetic strategy based on RNAs, a technology similar to the vaccines of modern vaccines of Moderna and Pfizer, pretty much, but used to epigenetically reprogram cells to rejuvenate and turn back their biological clock. So that's me in a nutshell. There's probably more, but I guess I can stop here.
Okay, great. Thank you. And Robert?
Yeah, thanks. So my name is Robert Zimmerman. I'm co-host here at the Longevity Biotech Show. I'm a researcher in an aging biology lab based in Quebec in Canada, and I have about 10 years of experience in bioinformatics and research software engineering. Okay, back over to you, Nathan.
Okay, great. So I guess my first question for Marco is really basic. What is Rubedo Life Sciences, and what are the kinds of therapies that you guys are trying to develop?
Sure. So let me maybe do a step back, because you mentioned correctly that Rubedo is a senolytic company, so developing therapeutics to target senescent cells. I would say Rubedo is not a senolytic company per se. So the idea behind Rubedo and the platform that we built, which is called Alembic, and maybe for the sake of interest, the all names Rubedo and so Alembic are names that they come from alchemy. And in an alchemic literature, the Rubedo is the last phase of the operation to realize the universal medicine, the elixir that gives you longevity and immortality in the mythology. And with the idea that really we wanted to develop therapeutics, so novel medicines that are customized for the biology of aging and for older people. And so to do that, we really wanted to make novel compounds. Technically, it's a novel composition of matter, so not really screen or find existing compounds, but design and make compounds that are made for this purpose. And why so important? It's because with age, things change, as we know. After a certain age, there is this exponential risk to develop, one or more age-related diseases. Right? Everything happens exponentially after a certain point, and in humans, it's typically around 60, 65, where there is this, inflection point, and then, there is every year a higher risk to develop a number of comorbidities. And there are reasons why behind that, which means they're changing biology, and so the patients are also different. As we age, an older person has a different metabolism. Drugs are metabolized differently. There is a lower tolerance to side effects and toxicity. So there are all things to take into consideration. And even more that, there is the fact that there are some rare cell types that slowly emerge and accumulate with age and eventually drive age-related diseases. Senescent cells is one of them, but there are others. So the idea behind Rubedo is how can we find the rare cells that drive age-related diseases? How can we really identify, create an ID, profiling them, and then how can we use this information to make drugs that are engineered to selectively target those cells and not the others? Right? In this way, we protect the healthy cells, we target the bad cells that are driving the aging process. So we solve these three problems in this way. We make drugs that are selective, that have a high efficacy and high tolerability and safety profile for aged people with age-related diseases. So that's what really Rubedo is about. And we can talk more about that and how we do that through our platform that we call Alembic. Again, another alchemic name. Alembic is the apparatus used in alchemy to make the medicine, this elixir. But it also means is an acronym for us that stays for algorithms for life-extending medicine using biology, informatics, and chemistry. And so I can tell more about that. But again, the idea is making drugs in different strategies. Let's say the platform makes sophisticated compounds such as prodrugs, complex prodrugs that can activate in certain cell types. And we have a similarity program, as you anticipated earlier, to make those compounds that can selectively target senescent cells. But again, senescent cells are heterogeneous, they're different. Depends of age, depends of disease, depends of tissue. So how can you do this pharmacologically, right? The idea is, we have beautiful data, for example, genetic models using animal models, right? But we cannot do a transgenic human to selectively target those. But so we came up with a strategy to make selective drugs that can have the same level of precision and safety as well. I'll pause here if you have any questions before I continue.
Okay, yeah, that's great. So, as I understand correctly now, that Rubedo has a Senelex program, but you guys are focused more generally on aging as well, just finding these special cells that drive aging, whether they're senescent cells or some other type of cell. So that's really interesting. You did mention the targeted nature, and I think this is really important because, as you know, in the senolytics industry, there has been a sort of high-profile failure of one senolytic drug company, and a lot of people are saying, okay, maybe the reason why that company failed was because they were too localized, like it was a local injection, and they had to be local because maybe the drug was too toxic to administer systemically. So this idea of selectively targeting senescent cells or other bad cells is really important in terms of how effective the drug can be. And so I'm wondering, maybe you could just walk us through how you do selective targeting. Like, what is the technology there? You mentioned something about prodrugs.
Yeah, and that's one approach, right? We invented several strategies, novel chemistry, for example, and the computational approach to address that. So let me kind of try to give a high-level view of the approach. As I said, everything starts with our discovery platform, Alembic, that is really a sophisticated, let's say, multi-dimension, multi-omic approach that really the idea is how can we look across multiple dimensions to see what's really emerging with age. So across multiple omic data sets, importantly, multiple ages, right? But then there is multiple tissues, multiple species, multiple disease models and patient populations to really triangulate what is emerging with age, what are those cells, and what is inside there, what can we target, what can we take advantage in designing certain chemical strategies. So Alembic is not just let's say, a bio and chemoinformatic approach that gives you an information, but is designed based on our unique chemistry approach to enable the generation, the development of these compounds. And an example, as you mentioned, are, for example, prodrugs. What are prodrugs? Are compounds, are molecules that are inactive, at least the way we design them. The idea is, let's make drugs that don't work by design. So we really are putting lots of effort in making drugs that don't work unless they find the right cells, right? And then they are activated into an active form. And if you need it, right? The idea behind those prodrugs or other strategies that we're using is, if you take, let's say, they talk about senescence and senolytics, right? Let's say you're young and healthy and you take a Rubedo prodrug and you don't have senescent cells, nothing happens. But if you have those cells, then you can trigger the activation of the prodrug selectively and proportionally to your age and disease and need in that sense. So there are multiple ways to do that. We definitely advanced a number of approaches. I can't disclose into many details, but for example, we released in preprint a manuscript. Now we're doing it's in the revision process for peer publication, but the preprint is available. And in that case, what we did is, again, that's a very simple prodrug. What we are developing actually therapeutically are more sophisticated than that. But in that case, we asked a simple question: is can we take a pan-cytotoxic compound? So a compound that is very toxic, it can kill senescent cells, but also other cells, and taking advantage of a known marker of senescence, which is beta-galactosidase, is an enzyme, right? It is expressed and enriched in senescent cells typically. And can we use this enzyme as a tool to activate our prodrug and in senescent cells that have high level of this enzyme? And the answer is yes. And so we proved this in vitro and in vivo. We tested this in geriatric mice, very, very old, 30 months or older, that might correspond in a geriatric person, maybe in the 80s or 90s. And we, where we proved that the compound becomes very well tolerated, right, compared to the unmodified version that is very toxic and kill even young animals. But instead, the prodrug can be used chronically, dosed repeatedly at high doses into very frail old mice that can tolerate that. But we also found that it was capable of in vitro, but also in vivo in animals, selectively clear those burden of senescent cells, resulting in restore of function. So the question there, we didn't want to see, can we delay aging? We wanted to ask, can we reverse some of this, age-related loss of function, such as cognitive functions? Those mice are kind of a dementia. They don't remember. They don't move. They have frailty. They're weaker. Their muscles are weaker. They don't regenerate as younger. And then after treating them, they tolerated well the treatment, but they also recover in muscle function, in stem cell functions, regenerative functions, in cognitive functions, and memory, and etc. So that was a proof of principle, right? And we definitely move way more advanced work and studies in the generation of our compounds. But that gives you the idea of, for example, a type of, in that case, simple prodrug using a very generic toxic compound, turning into a selective senolytic compound. And that was the first proof of principle that we did back in the days.
Yeah, got it. Yeah, that's really cool. Yeah, so anybody who's interested in reading more about the paper Marco's talking about, it's you can find it on their website actually in the publication section. It's a preprint currently. Yeah, very cool results. So what about clinical development? So on your website, there is like a clinical program that you guys are developing right now for, I believe, IPF or some sort of pulmonary fibrosis. Can you tell us what's in store for Rubedo in terms of trying to try some of these compounds in and therapies in human patients?
Yeah, we are working to advance. We're in a preclinical stage, right? I want to make sure that we are not ready to go first in human, but we're gearing up for that. So we lead compounds and we optimized, and we definitely generated superior lead compounds that we are advancing in our studies to move into IND. And if everything goes well, hopefully we can go in a in a clinical phase one trial in the next future. I'm hesitant in setting dates or estimates of times because science is also unpredictable, right? So we are approaching this extremely carefully. We don't want to rush in the sense that we want to make sure that all the studies, both tolerability, safety, and efficacy, are well-rounded and careful to be as translational as possible and predictive as possible before going to humans. So if more work is required, we want to do that rather than try to be to rushing into clinical stage at this point. But we're moving quickly forward, and as you said, yes, idiopathic pulmonary fibrosis is our probably most advanced program, but we have others that we are developing internally with a portfolio of compounds that we generated, and we are continuously expanding it in our the discovery effort.
Yeah, great. I guess maybe we can maybe go back in time and talk about the backstory behind the company. So, what was it that made you decide to start Rubedo Life Sciences? Like, what was the catalyst at the time, right before you started the company?
Well, depends how back in time you want to go. I guess that I had, in a way, for me, this the whole search and exploration around aging and how we can change that, improve that, is an inspiration of mine. It's probably a personal mission that I have that started at a super young age. I was a little kid when I wanted to understand aging, and I have this inspiration that they can be changed, and chasing this idea of naively at that age immortality. I'm less naive now, but I started very early. I had my first lab. I put it together at the age of 7. I was probably 6 when I said I wanted to change aging, and I have scientists and physicians in the family enough that I could like steal and borrow gears, chemistry gears, the microscope, and those kind of things. So I've been in this path of scientific and philosophical, and not just scientific exploration of for my whole life. And so my academic career and now more in this biotech development is always aimed for this concept of can we understand how aging works, and can we translate this into ways that can be changed to improve quality of life, and most importantly is personal, is can I do something and give back to society of my effort, which to me translates into can I make therapies, can I make new medicines that can benefit people's life. And maybe a point in general about this longevity space, that sometimes I hear comments of, well, yes, there is an aging population, but there is a growing population. This is already becoming a burden, right, for the planet. Do we need to have people living longer? And I said, well, that's not the point. The point for me, and I think in general, is can we make people living healthier for longer? So giving more life to their more life to their years, right? Rather than more years to their life. And why this is so important? Well, for example, the economical, socio-economical burden of sustaining an aging population, if we can postpone in time the onset of age-related diseases, would alleviate enormously the cost and healthcare costs, for example. It would also add a wiser population, a generation that can still contribute and happily be active, involved in the society from the older people that are healthier and younger in that sense. So those are all personal drivers to me. And Rubedo specifically, I found in not just the technology and the approach that we have that, I thought that I've seen many strategies and approaches, but I thought that that was really the chances to deliver, to execute those and make a new generation of therapeutics that can really target age-related diseases first and aging as a whole, aspirationally in the future. But also the team that we put together that definitely is a uniquely positioned with leaders, legendary names in the fields that join Rubedo to really join me in this mission. So those are all maybe a long answer to your question, but those are all reasons or drivers that personally made me choosing in leaving academia and focus full time in Rubedo to move forward in our mission, and hopefully we can achieve this goal soon.
Yeah, great. I think I'd like to focus more on this you mentioned you left academia to work on this problem of aging, starting biotech company, Rubedo Life Science, and some other companies as well. Um, Maybe I'd like to ask you just what are the sort of things that um, academics should know about starting a biotech company, right? Like generally speaking, I think a lot of graduate students or postdocs or whatever, they're trained to become academics, right? But then a lot of them transition into starting their own biotech startups or whatever. And I'm wondering, what sort of things should they learn, right, to be prepared for this sort of startup endeavor?
Hmm. That's a complicated question because there are multiple answers, I guess. Well, it depends. First of all, I think what kind of role and position someone who wants to transition from academia to, let's say, a corporate, let's say, ecosystem is looking for, right? Because this one thing being, for example, the CEO of a company versus being a scientist versus being business development or so that already changes, and I think it's important to be prepared. I think what is super important is finding the right people to work, having the right mentors, and because there are lots of things that needs to be learned, and I also take the time to do that. Right, academia is a different has different goals. You work in this idea of diving down into expanding knowledge, understanding basic questions, and there is a mechanics around that is very different into what do you need to actually turn an information into actually something real, and there are so many other layers that you need to take into consideration. A discovery in basic science, for example, it could be incredibly important and exciting, but it might not have a real value in terms of practicality or can be translated into something real. And so I think I wouldn't jump too quickly into, hey, this is great, let's do something out of this, because to de-risk since it's a very complex endeavor. So to de-risk the failure and maximize chances of success, I think it's important that there is the right expertise, the right knowledge, and it takes time to build that understanding on and moving very carefully from one stage to the next and go step by step. So typically academia, in my experience, doesn't really give you all the required tools. I think it's good to, you know, work with someone who has, you know, veterans maybe from that have been working in the specific space that you want to transition into, and and learn and making sure that you know what you're doing. But there is value as an academic also, because you know the way of thinking out of the box and come up with new solutions, that's invaluable, but it needs to be supported in a framework that enables the the execution of an idea into something that is real. For example, in therapeutics, and there are many layers of complexity in between. So I'd say the best is that don't do this by yourself. Don't try to reinvent the wheel, and seek for the strongest team as possible that can really fill the gap and that you can learn from if you don't have that experience, which takes time, takes a long time.
Yeah, definitely. And you mentioned having the right mentors, right? So maybe you can speak to that. Like, what kind of mentor would be ideal for like a young founder who's starting off, who maybe wants to start like a longevity biotech company, and how can they find these mentors?
Well, um, that's it's a bit of luck, I guess. But it's also, I think, the reputation. It's important. It's one of the most important things. So do everything you do at best as you can at any stage. Right? If you are a graduate student, try to do your work at best as you can, and working with the best people as possible, because that it kind of opens the door for what's next, and attracts other talents and that you can work with. Finding the right mentor, it's not easy, definitely, and you need to be exposed as much as possible to many people in the space. Now, if you're saying what's the best mentor to start a biotech company, it's hard to say. That its personality takes a lot, but certainly you want to see what's a track record of a good mentor. Is this an established person who proved already successfully that that it did before what you want to do, maybe multiple times? What's the reputation of this person, and what are the accomplishments? And certainly, is there a cultural fit with you? I think these are all important aspects that you need to take into consideration. And there is not only one, right? You need multiple mentors that you can learn, especially if you need to start a company, right? There is a scientific aspects, there is a financial, there is entrepreneurial, there is a business aspect, legal. So there are many components that I'd say you should try to always work with the best, because and the other good thing is typically people who succeeded well, they want to pass this knowledge. They want to see others following the same path, and be mentors in that sense. So, you know, I think they're out there if you look for them.
Great, yeah. So before we open up for questions from the audience, maybe I'll just give this time to you to maybe if there's something that we can help you, or are you hiring, or if you're looking for investors or something like that, maybe I'll just open up the floor to you. I know you guys somewhat recently raised your seed round from Khosla Ventures, which is incredible. Congratulations on that. But
Thank you.
yeah, if there's anything that you wanted to say to the audience or people listening to the podcast later, yeah, here's your chance.
Oh, well, thank you for the chance. Actually, yes, we closed not too long ago our seed round with Khosla. We are actually getting up for our Series A. and it's happening very soon. So we are ready to move to the next stage. We definitely reached very quickly that inflection point that at this point we are ready to move to the next stage. And so if there is interest and people want to hear our story, that we should be able, we expect to start the fundraise soon, really soon. So if anyone is interested, and please, you can reach out to me. I'll add to the list, and when time is ready, I'll open up invitations so people can book a time on our calendar and join the round of presentations for our Series A, which is going to happen soon. So feel free to reach out to me at my email, as Marco Quarta at rubedolife.com. Happy to connect and have conversations if there is interest about. That's in terms of investment round for the Series A. We are also very open to partnership. So if there is any interest for discussing new exploratory programs in partnership or in general, again, reach out to me or to partners at rubedolife.com. And hiring, well, it's on our website. We always have some open positions I think we have 2-3 positions open right now. So you can look on LinkedIn or again on the website and to see open opportunities. And I think we are looking for a couple of scientists and senior scientists and lead scientist position level right now.
Okay, great. Yeah. So anybody in the audience or listening at home, Marco mentioned that they are raising a Series A round very soon. So if you're interested in this or know someone who might be interested in investing in a company that's also backed by Khosla Ventures, doing some really great stuff, yeah, definitely reach out to him. And he also mentioned that they're hiring. They have some positions on their website. I also see that there's an internship, so other people who are looking for possibly an internship can take a look at that listing on their webpage as well. Okay, cool. So we'll transition now to Robert if you have any other questions. But if you don't have any other questions, we can also just go to the audience. What's your call, Robert?
Yeah, I mean, people can come up here and we can start the questionings. I have a few here that might be of interest to some other people also. So, Marco, you were talking about how you got started in the field and you had a very long-term interest in this and so on. So a question that's interesting to me and might be interesting to a lot of other people, I think, especially those who are not as familiar with the field of aging biology. What references, like textbooks, monographs, etc., would you recommend, or websites, or anything for that matter, would you recommend to people that are interested in learning about the state of the art of the field to get started and learn as solidly as possible what's going on?
It's an interesting question. Well, there are initiatives like yours. There are a number of blog and other initiatives that I think are very valuable that people can look at. Let me think on top of my head, like Fight Aging, for example. You can read, the authors there are always very very active and in adding news and reports and investigation around what's going on. Let me see. We have, it's. I don't think there is a necessarily one comprehensive aspect. My best suggestion is to be updated to scientific literature, right? Looking at some of the top tier journals. And recently, like Nature Publishing Group, for example, open up Nature Aging. For example, that's a great way to read about that and be up to speed. And but in general, I think I would try to look carefully on the scientific literature rather than try to to be too general in that sense. Um,
Maybe it would help if I rephrase it a little bit. Are there any books or papers or anything, or any comments or anything like this that particularly influenced your
hmm,
understanding of the biology of aging?
Probably not. um, I think it's a complex phenomenon. So there is not like a review, a journal, a paper, a book. I think it's a collection of all of it, and I constantly, read and explore things that it's hard for me to identify the one specific source of inspiration. That's why you hear me be struggling here and giving potentially a good answer. I probably don't have it. Sorry.
No, fair enough. It is a complex field for sure. One other question, then I guess we can go to Juliana here. So you were presenting a couple of years ago at a Foresight Institute event on an idea called the SymBio Watch, a wearable for longevity. And I confess I still haven't actually watched your presentation there in full, but it's a very interesting concept, and it certainly relates to the hardware side of things. So do you have any maybe brief description of this and some thoughts on people that are interested in contributing to the longevity biotech activity from a hardware perspective?
Oh, that's an interesting question. Yeah, that was an interesting exercise that, and actually a great event at Foresight. And yeah, I mean, I as a also bioengineer, I been actively involved in my past in initiatives of like biomedical devices or by my first company actually that I started back in Italy when I was doing my PhD, and that was I started by participating to a business plan competition. It's a national competition called Start Cup back in the days, and which was a great learning experience, right? You are exposed to, you get a business angel and you're trained, you learn how to do a business plan, those kind of things. But we happened to win and we got the money and we started a company, which is called the Wetware Concept. And the company is still actually up and running down there. The reason I'm saying is that in that case, we were really combining hardware and biology together. Wetware is a name that I took from cyberpunk literature and is kind of the evolution from hardware, software, and wetware is, the biological matter that can process information. So we were doing there is using semiconductor chips combined with engineered muscle cells or neurons. That was a while ago, almost 20 years ago. And to create devices that can, detect biological events and use as biosensors or potentially invasively implanted for other applications. So that was something that we did back in the days. Then the company took a different direction. We did a joint venture and we started to make more lab on chip kind of things. But the SynBio Watch, the idea that you mentioned, I think there is a tremendous opportunity in that sense, especially because the field matured since then to a point of complexity that I think there are tremendous business opportunities and ideas of how can we use like devices in the longevity therapeutic space. Well, in for example, developing like a non-invasive or minimally invasive diagnostic devices that they can inform, read, or predict um, markers, biomarkers of aging and disease longitudinally, so keeping track of what you're doing and giving you predictions of your health state or how, interventions that you could do to change how you're like aging, maybe your lifestyles, or you are exposed to factors that are actually accelerating your aging, biological age, not chronological age. And so you might need to prevent this before you reach that, you're accelerating maybe your inflection point, or the other way around. Can you slow it down? And that could, be used in also as a beyond that therapeutically, to as developing diagnostic kits that can be used. They're really important in the aging field. I know that NIH and NIA in particular is very interested in that. So that, for example, could be a very good valuable proposition in the longevity therapeutic space, longevity space in general. How can we develop devices? And now, there are, multi-omics. It's becoming cheap. There are ways to, accelerate analysis through AI, machine learning, and a number of other algorithms that can really use to kind of decode a massive amount of information that we can collect or we can track over time, and maybe on integrated way, sharing information with others, building platforms around that. So there is definitely opportunity to combine, again, hardware, software, and maybe, to go back to my initial point, wetware, so sort of a hybrid interfaces or devices that can collect biological information in real time or over time to predict how the aging, your biological aging is going, is changing, or potentially reverted as well.
Yeah, one final thing, because it wasn't mentioned earlier, is your company somehow potentially open to investigations involving drug delivery technology? Like, for example, the mRNA vaccines for coronavirus are using lipid nanoparticles. Excuse me. If such a thing were available, would that, to your company, would that make things better or easier, or improve the technology in some way?
Well, so at Rubedo, our platform makes small molecules. Those prodrugs are based on small molecules. So like you don't need any specific system to deliver these drugs. So that's another strength of the approach, right? We take advantage of an established pharmacological development process based on small molecules, well known. And so it can either be like a pill, it could be another way to administer a drug, but are small molecules. So there is not really a major issue, in making formulations for that, because this is an established field. Now, there are other areas, especially in biologics, that this is still an open question. And for example, I mentioned another company that I co-founded and I'm involved with, which is Turn Biotechnologies. In that case, it's based on an RNA technology, so biologics, and the delivery is very important. So in that context, we're developing novel delivery strategies to combine, to effectively deliver this RNA-based therapeutics, which recently, through the pandemic, we learned how is important in, for example, development of vaccines, like you mentioned, right? Using nanolipoparticles to enable the delivery of those RNAs in the context of a vaccine, for example. So yeah, new delivery approaches, especially in biologics, for a number of different longevity therapeutics that are currently in development with multiple companies and more to come, is certainly a very powerful and very valuable and needed, and there's an unmet need in certain areas, which I encourage people to look into because it would be also a good business opportunity as well.
All right, back to you, Nathan.
Okay. Yeah. So just to reset the room, we have Marco Quarta here, the founder of Rubedo Life Sciences. And just for anybody who's coming up to the stage, just a reminder, if you come up here to ask a question, it means you consent to us using your audio in the recording and your profile photo in the video that we upload to YouTube. So let's go on to Juliana. What's your question?
Thank you very much, Nathan and Robert. And thank you, Marco, for the talk. It's really interesting. So I have actually two questions. So the first is, what was the rationale behind the choice of IPF and COPD as your lead disease indications? Because being an orphan disease brings several regulatory advantages that streamline the process. But is there specific evidence in the literature indicating that a systemic depletion of senescent cells would be enough to counteract IPF and reverse the disease scenario? And my second question is, although it's actually pretty early for that, do you have an idea of what will be your strategy for clinical trials in terms of endpoints to be measured?
Great question. Thank you, Juliana. So to the first point, yes, there was a careful work around which indication we should go first, right? So Rubedo is not a, let's say, respiratory pulmonary disease type company only. It is not an oncology company, but we could go after all of that. But that poses also, and that's a typical question for companies like ours, going after, mechanisms of aging, which is our master regulators of multiple diseases. Then say, so what do you want to target first? So we carefully thought about that. And IPF, yes, as you said, yes, is an orphan disease status, and there are maybe certain advantages in terms of clinical strategy. And but beyond that, it's really that the role of senescence in IPF is becoming more and more clear to be a driver, a very important factor that could really resolve or have a very strong therapeutic capacity in this patient population. We partner up with the Cedars-Sinai Medical Center at LA, and Professor Hogebaum is a professor in the respiratory diseases, and it's been working for 25, 30 years in IPF and cellular senescence, and he joined forces with us. And the field is coming to the growing conviction that the role of cellular senescence in APF plays a critical role. So we decided that we wanted to hopefully bring to the clinic and a first treatment that can really benefit the patient population. And so we decided to prioritize IPF towards other programs that we initiated internally. So yeah, we definitely strongly believe that targeting senescence for APF could be really important. Now the point is how to target this, and it's not just necessarily that a senolytic approach might be resolutive as we think. So that needs to be done very carefully, and that's what we are really working hard to develop our program for that. And to the second point, yes, we have a clinical strategy to do that. We have a team on product and clinical development that we're working very carefully to identify the right way to go to the clinic, which patient population endpoint stratification, how to address that. But these are still, it's too early to maybe talk this into too many details, but you have a clear understanding, at least a clear idea of how we plan to do that.
Awesome, super interesting. Thank you very much.
Of course.
Okay, yeah. If we don't have any other questions, I have a ton of questions that I can ask Marco. Um, so back to our point about unity biotechnology, right? And this idea of localized administration or systematic um, administration of senolytic drugs, right? So there's this idea that to be able to really, I guess, ameliorate the effects of, the SASP, right, the inflammatory and like, pro-fibrotic effects of senescent cells, you have to go systemic. Do you think that's true, or is there a way to do it locally, or yeah? So do you think we have to go systemic to be able to address the problem?
Well, so I think, senescence biology is also more complex than we think, and we are learning this every day, right? So I think we need a lot of different efforts to develop multiple senolytic strategies. It's a little bit like oncology. It's not that there is a cure for cancer. There are many types of cancer, so senescence can be different. It's not that because there is some evidence with some existing compound, for example, the cocktail D plus Q, that in animals showed some efficacy and can be used, and they're like suggesting in some clinical trials run at the Mayo Clinic that there is efficacy, but that's not going to be a cure for everything, maybe even for anything necessarily, without being carefully developed. And same is in general. Well, how do we target the biology of senescence as a therapeutic opportunity? So that's where there are multiple ways, multiple strategies, and it's not that simple. So different compounds, which pathways, how you target senescence, in which cell types, in which tissue. It might. So to your point, there might be. There are possibly indications, clinical indications, that a local administration could be advantageous. It is true that if we think in the context of an aging process where, if we can simplify thinking, or you keep accumulating senescent cells, overall increase chronic inflammation overall, so you need to clear this systemically to change significantly the whole body status. But then you need to look specifically in a disease, right? There might be a specific role of senescence and accumulation of senescence in a certain disease, in certain indication, where a local administration could be advantageous. And so I think what the initial thinking at Unity in going after osteoarthritis, for example, and using those, yeah, they may have limitations for systemic administration on their compounds. But the idea of using some privileged tissues and do local clearance of senescent cells, it it makes sense in certain ways. And now they're targeting macular degeneration. They're moving in phase 2. I think there is a good chance that that actually could show efficacy. I think it's hard to say why that didn't work for osteoarthritis, and I wouldn't simplify in saying it's because the senolytic approach didn't work in that case, right? When you run experiments in animals, you can run many, many conditions and try to dissect and understand how things work. Drug doesn't work just, you take it. How do you take it? What's the dose? When do you take it? You interfere with biological events. This is all, developing a drug is understanding all the pharmacology around that. It's not obvious. That's why all the preclinical studies should be as careful as possible, because when you go into humans, you have one chance. Now, those are very expensive and complex behaviors. So if you choose the wrong endpoints, or if you predict them, you try to model as best as possible. But then when you run this human experiment, this is what a clinical trial is, and you don't see results, doesn't mean that it didn't work. It means that you might not have enough statistical power to demonstrate your hypothesis, even if maybe the drug is working, you missed the right dose, you missed the right endpoint, and you should go back and do it again and change the conditions. Not everybody has the capacity to do that, right? There's a huge cost and complexity to do that. So I think that the clinical trial for osteoarthritis that Unity tried in phase 2, unfortunately didn't work, but there are a number of reasons that that could be not necessarily that local treatments failed, maybe using a different dose, different multiple doses, or looking at the same endpoints at earlier or in a different ways could have actually led to success. We just don't know that.
Yeah, totally. I agree. Um, Maybe I have one more question since we have a
Sure,
little bit of time.
sure.
Um, so, from your point of view in the longevity industry, what are the low-hanging fruit for other potential, I guess, startups to tackle? Or maybe even if it's not like a specific therapeutic area, maybe there's some sort of infrastructure or some project that's required to help accelerate progress in the field. So, like for example, I asked this question to Kristen Fortney of BioAge Labs, and she mentioned the whole problem with uh, old mice, right? To do a lot of these studies on old mice, it's difficult to get these mice, it's very expensive, et cetera, et cetera. But I was just wondering if you had any ideas about like, hey, maybe this area or therapeutic area is underserved in terms of new startups being formed to tackle it, or some sort of infrastructure that's required to help accelerate progress.
Hmm. Yeah, that's a good question too. Uh, the low-hanging fruits. There are on one side there are many. There are so many unmet clinical need in the age-related diseases indications. Interestingly, they were not the low-hanging fruits until now, and because that's why, let's say, the pharmacological development of the past 50 years did not address those because we didn't have the capacity to go after those. Now we are reaching those that are in the context of targeting aging. We go at the root of some of those degenerative processes, chronic conditions, and they're starting to become, quote unquote, long-hanging fruits. But of course, it's complex to go after them. Um, and I think, you need the right biology and you need the right model for that. As you mentioned, like using aged mice is expensive and is complex. I would add that it's not necessarily the best model. It's better. You look at an aging environment, and it's definitely you need aging into your equation to make good prediction. Like what are the predictors for potential clinical translations. But animal models are not human, so learning how to cure a mouse is not does not going to help really or translate in how we can cure a human. So that needs to be clear. So, for example, finding better models that can better inform the biology of aging in humans clinically, that would be a tremendous tool, to develop, and it could have it's a product itself. Many other companies could benefit and maybe use and work in partnering with whoever has good models to do that, or that can enable sort of studies or understanding aging in humans better and faster, and so you can test therapies there that are more reliable than using animals, especially rodents that are not that similar to humans for many ways. Um, and at the same time, if you have that, you now have also a competitive advantage in testing therapeutics internally. And possibly you have models that can work better in one indication or in the other. So how to think about this? Well, you could think about what are all the hallmarks of age that we know that are most established, and how can you best use them to develop models that can be as translatable as possible, preclinical models, or even human or human-like or human data that you can use to model potential therapeutic outcomes. So I'm not giving you a specific answer, but I think I'm giving you potential inspirations for ideas.
Yeah, great, great. Thank you for those ideas. Yeah, I think having some better type of preclinical model would definitely help. There's a lot of talk about like organoids, organ on a chip, maybe some other approaches. You could also try like non-human primates. A lot of people are looking into that. I mean, they already have that, but it's there are different models like marmosets or whatever that could be interesting. Yeah, I guess uh, I want to be mindful of your time now since it's 9:04, a little past. I just want to thank you again, Marco, for coming onto the show. It was really great to talk to you and very fascinating conversation talking about all the exciting developments that are going on at Rubedo Life Sciences. And I just want to just reiterate to everybody listening that Rubedo Life Sciences is going to be raising a Series A at some point in the very near future. So if you're interested in that or know somebody who might be interested in investing, then definitely reach out to them. There's also many open positions on their website. So if you go to rubedolife.com, they have some positions for people who are looking to join an anti-aging company. And they also have some internships. So with that, I think we'll just come to a close here. And yeah, just thank you again, Marco. It was great having you on the show.
Well, thank you, Nathan. Thank you, Robert. It was a pleasure. I really enjoyed conversations, and I wish we had more time, but hope that we'll have another in the future. And thank you again for the invitation. And please continue with this show. It's definitely great and very valuable for the space.
Thank you so much. Yeah, it was our pleasure having you.
Likewise, being here. Thank you.
All right, thank you. Take care.
Take care. Bye, everyone.